So in last video we were done with the role of nucleus in the development. Today we are going with the role of cytoplasm. In development, so first thing you should always be known about what the concept we are going to study. That's about role of cytoplasm. So cytoplasm is simply a fluid-like material present in the cell, every cell. This because what we'll see is that a cell, if this is the structure cell which have a nucleus in the middle, then we'll have mitochondria, vacuole, other like things, and of course they need so that they could not do friction, they could not like cytoplasm is simply a watery substance which provide them a lot of things like to not paste and live in the cytoplasm. Like simply it's a fluid which help in so many things. Again, the cytoplasm is divided into two parts. Basically cytoplasm is not the topic, but before we continue you should always be known. So cytoplasm is having two parts, cytosol and cytogel. So when comes cytosol, what are these and what this concept mean? So cytosol, this remember these words. Cyto word is used because it's about cytoplasm, and gel and sol, what does this mean? This mean if I just make a structure, here comes the structure. What you'll see when I talk about the cytoplasm, so here in the edges, in near the border, you'll see the cytoplasm is quite thick. This means a gel like structure, so that's why we just term the middle part called, not middle but the end part called the gel. When comes about cytosol, so cytosol is simply near the, if here's the nucleus, so near the nucleus the fluid that's called as cytosol. So I'm sure it will be clear then. The cytoplasmic component, this means this is quite important in zygote formation, because even if the all organs are formed, not all organs but all nerves, these things are formed by nucleus, so they need a place where they could stay. So place like what? So for their place they have called as cytoplasm. So I'm sure it could be clear. Then comes, okay, after that we have cytoplasm, which means they have different components. Cytoplasmic component contain different morphological, contain and different morphogenic. Morphogenic is simply having, morpho means shape, so form and structure called as morphogenic. Then this is even essential for cell differentiation. Cell differentiation is a technique, so cytoplasm is also important for essential for cell essential for cell differentiation. Then they are present within the blastomere. So within the blastomere, this means when we just make the structure, this is the zygote is also called as fertilized egg, and it is formed by the fertilization or the mixture, not mixture but the fusion of ovum and sperm. Then comes the second part, which is about having division of two sides. This is called as mitosis or cleavage process. Mitosis and cleavage also have a difference. Mitosis simply occur in both plant and animal, whereas cleavage only occur in the animals. Then comes the blastomere, which means therefore divided. So the first one, every single cell called as a blastomere. So this means they are cell differentiation and they are present within the blastomere. This means cytoplasm would be in these three. After that it become more fused, they make morula, which is about 60 cells combined. And then we have taken an example which is SCDN. Okay, sorry. Then we have just taken example of SCDN. What does this just mean? Our cytoplasm have different colors. This is an example. Then cytoplasm, basically in our book that's written something more, like they have taken an example of frog, but for now we need to discuss what is mainly the purpose, right? So we of course we'll understand frog, but it won't clear our main concept, how is used in development. That's why I'm just making you clear these things. So our cytoplasm, which have 5 different colors. If you're thinking we only have seen white color, you are wrong. Yes, I was also wondering like that, but not now. 5 different cytoplasm color. So then comes, okay, after this means when you just see clear cytoplasm, this means quite transparent or white. Clear cytoplasm, this means they have produced larval, or simply they are at the larval, or they are epidermal cells, simply outer cells simply. Then comes the second part when we talk about the yellow, which second one is yellow cytoplasm. When we just talk about the yellow cytoplasm, this means they are present in the muscle cells. When we talk about the gray, cytoplasm, that's called as present gray vegetable cytoplasm. If I'm not wrong, this means they are present in the gut. This means our digestive tract. When we talk about, we have differentiation, we call this as the vegetative or vegetable cytoplasm, and here comes gray equatorial cytoplasm. So this means this is present in what is present in notochord and in our neural tube as well. We discussed neural tube in quite detail in last few videos, you can check that. So I'm sure it will be clear till here we have our topic. So now let's see in the diagram, let's see in the book what's written, what's the importance and everything. So yes, picture I've gotten blur, I'll just need to edit again. Thank you, wait for a while. So here you can see role of cytoplasm in the development. First thing, the role of cytoplasm in controlling process of development. This may it help in controlling and the process of development, which have been revealed through experiment on frog embryo. So we have done an experiment on frog embryo. So let's see. So before fertilization, this means this means there is no any formation of fertilized egg or zygote. The unfertilized egg has a pigmented upper cytoplasmic half and a yolk lower half. So this means we have not fused them, but here's the cell. So what we see, when the upper fertilized egg, this is here, which is pigmented cytoplasmic, and the lower, which is yolk like, this means the yellow part like yolk like. What's more written after fertilization, this means it's a clear. Here we can see control egg. Here's a not result, but the first thing, here comes this part. Then we see after fertilization what happens. So a gray crescent forms opposite to the point where the sperm nucleus enters the egg. So the when nucleus enters the egg, they form a gray scent like structure. Gray scent, which is here this middle one, gray scent is formed when the sperm get fused. This gray scent is created when some pigments in the cytoplasm shift upward. So this is present because when some pigment shift upward and leave a crescent shaped area, they will leave this area and they will form that's why they form a color which is gray crescent. I hope it will be clear. Then what's written more, normal tadpole. Okay, here during the first cleavage, when the first cleavage happens, this means division. Cleavage is a division of mitosis. So the zygote divides vertically. This means they are going to divide vertically, like vertical is this one, sorry, vertically. Then and each daughter cells receive a half of the crescent. So if this is the cell and here we have a crescent like structure, so they get divided like this. So half crescent get to them and half to them. I hope it will be clear. Then interestingly, if these daughter cells separate carefully, each of them develops into a normal tadpole larva. So if they both develops or divide equally, so they form a normal tadpole larva. Tadpole is simply the baby of frog called as tadpole. If I'm not, yes, tadpole is there. Then in the experiment, which is created by Hans Sparman in 1930, which was altered the normal to the first cleavage, as a result daughter cells receive entire gray crescent. So when division occurred, so this means they are telling the entire gray crescent is received by the daughter cells and did not receive, and the while other did not receive any crescent. So both of these cells received the entire gray crescent, developed in a complete tadpole larva, while the non-crescent daughter cells failed to develop properly. So this experiment highlight that even though both gray crescent in the cytoplasm has significant effect on a gene expression and subsequent development. So they are mentioning if equally divided, so then the tadpole structure would be formed. If not, so what they are doing, the daughter cells is getting gray crescent entirely, which is not of course good. So here you can see normal, here this is formed. But when we've taken this one experimental egg, which is side view, so this means gray crescent is here, and here comes the middle part, which is the thread like structure would be formed. So one you see called as belly piece, between middle one, here comes the normal one. So in normal we will see gray scent, but in this one we cannot. So I'm sure it will be clear. Thank you for watching. Bye bye.